Right, in this video, we're going to look at the control and the regulation of the cell cycle. How does a cell know when it is supposed to divide? Well, it turns out that it depends upon the cell type, that not all cells will divide. And the probability of whether a cell will divide or not comes down to what type of cell it is, the conditions within the cell, so the intracellular conditions, and the conditions outside of the cell, so the extracellular conditions. So different, like I said, different cell types will divide at different frequencies. Human skin cells divide frequently. Human hepatocytes or liver cells can divide, but they only do so when the liver is wounded. And finally, nerve and muscle cells in mature humans will never divide. So let's take a look at this regulation. Let's take a look at the decisions, the various decision points that a cell will go through to determine whether or not it's going to divide. And it's all controlled by the cell cycle control system, which is not unlike the control system of a washing machine, in that there are various checkpoints that when reached, that washing machine needs to determine, okay, are we ready to progress to the rinse and spin? Okay, so it gets to this particular point and it asks itself, have we been agitating it enough with the soap? If so, go ahead and continue on to the rinse and spin. If not, continue agitating, continue mixing the clothes with the soap. And then it gets to the point where, okay, we've been rinsing. Are we ready to spin? That's another checkpoint. Have we drained the basin? If not, then we continue to rinse and we continue to drain. If it's drained, then we can go into the spin. And so each one of these various points in this control system acts as a checkpoint. And that's the same thing that happens with the cell cycle. There are three important checkpoints. There's the G1 checkpoint, the G2 checkpoint, and the M checkpoint. And at each checkpoint, there are questions that are asked. Are all of the chromosomes attached to kinetochore microtubules? If so, proceed. If not, do not proceed. So there are signals that then the cell is going to receive to answer these questions, to answer these yes or no questions. And these signals can be broken down into two types. Intracellular signals can also be called cell surveillance. Those are signals that come from within the cell. They report upon the internal state of the cell. Is DNA replicated? Have the sister chromatids separated? Right? These are the sorts of events that these cell surveillance signals are going to be monitoring. And then there's extracellular signals. These are signals that come from outside the cell. These are oftentimes inductive signals, so growth factors, for example, that are received, right? So here's our growth factor. It's a signal that's been secreted by a neighboring cell that's going to be received by the receptor of the target cell. It's going to lead to a signal transduction pathway, and in this case, the cellular response is going to be a go signal to tell the cell to, yes, go ahead and go through mitosis. So let's take a look at each one of these three checkpoints. The first is called the G1 checkpoint, or sometimes called the restriction point. Now, if at the G1 checkpoint here, right, if there is a stop signal, then the cell goes out of G1 and into something called G0, and this can be thought of as the working state of the cell, or the working phase of the cell. Most cells who are simply existing and doing their jobs exist in the G0 phase of the cell cycle. And it's only when an internal or an external signal has been received that says, go, we are going to divide. It's brought out of G0 back into G1, and then it passes through G1 into the S phase where DNA replication is going to occur. Let's take a look at what a signal might look like here at the G1 checkpoint. An example is an external signal called a platelet-derived growth factor. And these are secreted from blood cells, and they are received by fibroblast cells that have a receptor that binds to this signal. It's a receptor tyrosine kinase. And so we can do an experiment here. We can take some fibroblast cells, we can chop them up, well, chop up the tissue, not the cells themselves, put them into a beaker, a flask here, shake it up, pour some of it onto into a solution that does not have the growth factor, place them into a solution that does have the growth factor, allow them to incubate, and the only place where you see cell division is the sample that has the growth factor. And you can see that we have lots of cell division that has occurred. So this is an example of a signal, an external signal at the G1 checkpoint that tells the cell, go ahead, proceed. Now, most of the time, if a cell is told to go through G1, pass through G1, then it probably will go all the way and eventually divide via mitosis. Let's take a look at the G2 checkpoint. That's this checkpoint here that's going to look to go from interphase and actually start the M phase, mitotic phase. And the G2 checkpoint is dependent upon an internal signal called the M phase promoting factor, MPF. And the M phase promoting factor is dependent upon two separate proteins that bind to one another. The cyclin-dependent kinase, so let's break that down. Cyclin-dependent kinase. Well, it's a kinase. We know it's going to be phosphorylating things. And it's cyclin-dependent. Aha! It's probably only going to be active if it's binding to cyclin. Well, what's cyclin? Ah, there's cyclin. So cyclin is our other protein that when bound to the cyclin-dependent kinase activates that kinase. Now let's take a look at cyclin. What might be special about cyclin? Cyclin cycles. Aha. If we take a look at the concentration of cyclin during the cell cycle, throughout the cell cycle, what we see is that its concentration goes in cycles, thus the word cyclin. So right around the end of G2, it reaches its maximum, or as G2 is progressing, it reaches its maximum concentration, and then it is quickly degraded. And then again, as we are going through G2 of the cell cycle, its concentration is increasing, and then it is degraded. So cyclin is called cyclin because its concentration goes throughout the cell cycle in cycles. And because its concentration, right, goes up and then down and then up and down, that means it's only going to be available right around G2 in the beginning of M to bind to the cyclin-dependent kinase, thereby making that active. So if we look at the activity of MPF, it mirrors the cycling concentration of cyclin. All right. So now we can take a look at how this acts as a good internal signal to bypass the G2 to M phase. So during G1, cyclin-dependent kinase is all alone. Cyclin is at its lowest concentration. It's been completely degraded. And it's only if that cell is going to go through mitosis that cyclin is going to be produced. It's produced at its highest level right here towards the end of the G2 phase. At that point, it's going to be binding to the cyclin-dependent kinase, activating it, and therefore that cyclin-dependent kinase is going to begin doing what it does. It phosphorylates things. What does it phosphorylate? It phosphorylates molecules necessary to break down the nuclear envelope. It phosphorylates molecules necessary to condense chromosomes. It phosphorylates molecules necessary to begin forming the mitotic spindle. All right, so the MPF is active. It phosphorylated all of those various proteins. We've begun the process of mitosis. We've started the mitotic phase of the cell cycle. And now we reached the M checkpoint. And the M checkpoint asks the question, are all chromosomes kinetochores connected to microtubules? So are all of the various chromosomes here, consisting each of 2 sister chromatids, are their centromeres and kinetochores bound to kinetochore microtubules? If the answer is no, then we don't pass through that M checkpoint. If the answer is yes, that means that metaphase has occurred properly and we are ready for anaphase to begin, and we pass through the M checkpoint. Let's take a look at some of the molecules that are involved in the M checkpoint. What we can see here is that sister chromatids are connected to one another by cohesin. So these green here, these proteins are called cohesin proteins. The blue ones here are called condensins, and that's one of the things that makes chromosomes very condensed during metaphase. But we're mostly interested in these cohesin proteins because they are connecting the 2 double-stranded sister chromatids together. Okay, and you can see they're a ring that links them together. Now, if all of the chromosomes are not connected to kinetochore microtubules, so the answer to that question is no, then these cohesin proteins are going to stay stable and linked and keep the 2 sister chromatids together. If, however, metaphase has occurred correctly and those microtubules are connected to each one of the chromosomes, then the answer to this question is yes. And then what happens is a particular protein is activated called the cohesin cleavage protein, symbolized by the scissors here. So the cohesin cleavage protein cleaves cohesin, as the name suggests. That allows the 2 sister chromatids to separate from one another, and that allows for anaphase to proceed. So that is the cell cycle. G1 checkpoint, G2 checkpoint, M checkpoint. Stop and go signals will then determine whether or not that cell is going to proceed through that particular checkpoint. And this makes sure that cells are only going to divide when they need to, and that when they do divide, that things happen properly before passing on to the next stage. Now, let's take a look at cancer. Cancer is a set of diseases. Anybody who said, I'm going to cure cancer with this one particular molecule doesn't understand that cancer is a set of diseases. Lots of various molecules are involved, and a single cure is very unlikely. Now, what happens here is cells divide without regulation, and the cause can be genetic. Can be mutations that have occurred within the cells that stop the ability to regulate the cell cycle, and even epigenetic changes in the way the DNA is packaged such that the normal control of gene expression is off. Some sources of genetic mutations like chemical carcinogens, cigarette smoke, X-rays, ultraviolet radiation from the sun. And then there are some viruses, like the human papillomavirus, can cause cancer. Sources of epigenetic modifications, well, those are a field of study that's being researched now. Now, what happens here is cancer cells, because of these mutations, don't properly respond to signals anymore, whether those are external or internal signals. And depending on the type of cancer that we're talking about, some cancer cells, the problem is that they no longer respond to stop signals. So there's just there's the signals are being put out there, stop, 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 but the cancer cells don't read them properly. Some cancer cells have hyperactive go signals. So it's not a matter of, hey, we're not hearing the stop signals anymore. The problem is we're making too much go signal. Go, 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 go. And we'll look at the two types of genes that are involved in those two types of cancer there. There are several different types of mutations that have to occur to a cell to become a malignant cancer cell. One of them is they become immortalized, right? And that's a mutation that reactivates telomerase. And so these cells can divide and divide and divide without stopping. If they were not to receive that mutation, they would be a benign tumor. They would divide, divide, divide until you have a tumor, but eventually they'd reach that Hayflick limit, stop dividing, and you've got this tumor, but may not be, it won't be cancerous, and may or may not cause problems depending on where that tumor is. HeLa cells from Henrietta Lacks. She died in 1951. She had cervical cancer. Doctors took a biopsy of that cancer and they found indeed these cells were immortalized. They would just, as long as they kept giving them nutrients, they would divide and divide and divide. And so researchers then are still using this cell line as a way to study human cells. They're so easy to keep going and keep dividing. Okay, let's take a look at genes that are susceptible to cancer-causing mutations. And we got two classes here. We've got the tumor-suppressor genes and proto-oncogenes. Now, tumor-suppressor genes. Now, I should say that both classes of genes are absolutely normal, everyday genes, and you and me, we all need these tumor-suppressor genes. And they are required for the normal inhibition of cell division. So basically, they're involved in stop signals, okay? And as long as they work properly, they will tell a signal to stop dividing and everything is great. However, if a mutation causes this gene, causes the protein encoded by the gene to no longer work, the cell can no longer give out a stop signal. So, for example, P53 is a very important protein within cells. And it is a protein that responds to UV light damage. So DNA is damaged by UV light. There are proteins that recognize that damage. They activate P53. P53 is a transcription factor, sits on the promoter of genes, and activates the transcription of genes that encode proteins that inhibit the cell cycle. Beautiful. That is our off, that is our stop signal. But if a mutation inactivates P53, then we have these proteins that recognize this DNA damage. They try to activate P53, but it doesn't work. And therefore, cells can't turn on stop signals. And so the cells just keep dividing. Proto-oncogenes are the opposite. Proto-oncogenes encode proteins that are involved in stimulating cell growth. They're involved in go signals, go, 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 telling the cell to divide. Okay, which means the first thing is, if we give a mutation to a proto-oncogene similar to the mutations we were talking about, tumor suppressors, basically mutations that break the proto-oncogene, that's not going to work, all right? Cells are no longer going to make a go signal, so cells aren't going to divide. That'll cause problems, but cancer won't be one of those problems. So what that means is only certain mutations are going to convert a I got to misspell there, proto-oncogene into an oncogene. Again, simply breaking the proto-oncogene won't do. It's got to be special mutations. So let's take a look. There's all kinds of possibilities here. Let's start at the top. So here is our proto-oncogene, and maybe normally it's packaged up in chromatin in such a way where it's not accessible to RNA polymerase. Well, maybe there's a mutation in a gene that encodes chromatin-modifying enzyme, such that this part of the chromatin isn't modified properly. It's too open, allowing the gene here to be accessible to all of the RNA polymerases, and now we have too much of this protein, too much of the go signal within a cell, just constantly saying go, 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 go. Another possibility is that here's our proto-oncogene. It's under the control of a specific regulatory sequence that within a cell says, nope, don't make it, don't make the protein. But there are mutations called translocations in which the gene will be moved to another part of the genome, and maybe it gets moved so now it's under the control of a new promoter, a more active promoter, a promoter that's attracting activators that are turning on this proto-oncogene and making way too much, way too much, way too much, and therefore, again, an excess of go signal causing the cell to divide with no regulation. Another type of mutation is the fact we have our proto-oncogene that we have what's called gene amplification. This is another type of mutation in which the gene is copied and exists 3 times within the genome. Each one of these given rise to the protein, and now we have too much of that protein again. Another mutation, here a mutation within the regulatory sequence of the proto-oncogene, within the normal regulatory sequence, such that causing new activators to bind there and making too much of that protein. Notice every single one of these examples, 1, 2, 3, 4 examples so far, we haven't changed the protein itself. The protein is the same. What we've changed is the amount of protein being made within the cell. Normal growth cell in excess, normal growth stimulating protein in excess, normal growth stimulating protein in excess. So we haven't mutated the protein itself yet. We've just simply made too much of it. Finally, the one example where we are changing the structure of the protein itself. So a point mutation here within the proto-oncogene itself turns it into an oncogene. It is now hyperactive. So maybe we've mutated a regulatory site within the protein such that it can no longer be turned off and it's constantly active. So here is an example of just that. There is a pathway, so here's a growth factor, right? This might be the platelet-derived growth factor, for example, that binds to our tyrosine receptor kinase, that when active, activates a G protein, so this RAS G protein, which then activates a series of proteins that turns on a series of genes that stimulates the cell cycle. Well, there are mutations in the gene that encodes the RAS protein that stops its ability to hydrolyze GTP. Remember, these G proteins, one of the important functions is that they can hydrolyze their own GTP, thus turning themselves off. And so the mutation in the gene that stops this ability from happening means that RAS is going to be turned on, thus turning on all of these go signals, go signals, go signals, even if there is no growth factor, even if there is no active receptor tyrosine kinase. So here's an example of a mutation that we see. That's an example right here where a mutation causes our oncogene, our proto-oncogene, to become an oncogene, to lead to a protein that is hyperactive, leading to the production of too much go signal. Now, cancer is a multi-step process it needs a single cell needs a series of mutations to occur. First is the initiation, mutation in an oncogene, tumor suppressor gene, and then promotion, so a mutation to a second copy of a tumor suppression, of a tumor suppressor. And then progression. So this progression is some of the more easy things to understand. There needs to be mutations maybe around the cell that leads to more vascularization, because as these cells are dividing and dividing and dividing, they're going to need a lot of oxygen. So they're going to need increased vascularization. That requires mutations that cause increases in vascularization. Now, in order for that cancer to become malignant, to spread, it needs mutations in cell adhesion genes. Genes responsible in those cells binding to, so for example, if we're talking about the colon, so colon cancer. Well, normally the colon cells stick to colon cells because they make cell adhesion proteins. However, if one of those cell adhesion, the genes that encodes a cell adhesion protein is mutated, then now those cells can become detached from the colon and spread elsewhere. And finally, reactivation of telomerase, a mutation that causes telomerase to be turned on again. Thus, not only will we get a tumor that just keeps growing and growing and growing. So now we have a tumor that keeps growing and growing and growing. It no longer it's being fed by plenty of vascular tissue, and it is no longer adhered to the tissue that it started at. It's beginning to become detached and moving and spreading to other parts of the body. And so the question here is, who gets cancer most often and why? Well, the older you get, the more likely it is that you're going to come down with a cancer. Why is it? Well, mutations are random. The more time you give these mutations to occur, the more likely they are to occur. So since a cancer requires a series of mutations, it just is natural that the older you get, the more likely you will come down with some kind of cancer. And then the question is, well, what if you inherited one of these mutations? What if you inherited a mutation that already it turns on telomerase? Okay, so maybe you don't have cancer in those cells, but it's now, instead of taking 3 hits, it's only going to take 2 mutations. And so if you've inherited something like this, then you are going to be more likely to develop that cancer and develop it earlier within your life. Okay, so those are genetic predispositions to cancer. Okay, so in the next lecture, we're going to talk about meiosis, another kind of cell division, but a cell division necessary for the production of gametes, of sperm and eggs.